Our class is starting, or our school, university is opening at 10 a.m. So our uh, lecture class is canceled this morning. The lab class will meet in the afternoon because the USM will be open. And so instead of having in-person class or Zoom class, I'm just recording this, and you're expected to watch this with as much attention as you would have if we were in class. And as we're going, write down your questions so you can email them or especially bring them into class next week. Riley Cott was supposed to come to our class today. Riley's going to come next Tuesday. Riley and I already have that plan set up. So we'll see Riley on next Tuesday. We had an in-class assignment scheduled for today. We're just not going to do it. We're going to skip it. What we're going to do is get out clay and physically work with it, and we'll to build genes, just like the clay model videos. And we'll talk about that, and we'll incorporate that into our course in some other way. Then let's just get into stuff to be doing this week. So we'll skip that. Riley's coming next class, and we're finishing reading chapter 1. I'm going to talk about that some today. I talk about it in a different way than the book, so you got to read the book and learn about it. But it's an overview, and if give me a chance to give an overview and introduce the course, I'm doing it differently than the author. And so for the first couple of chapters, it's like that, but then we'll be paralleling our chapters more directly. Okay, and then we'll get through that, and we'll go on to chapter 4. And after 4, we're going to look at chapter 9. And chapter 9 is really about the clay model videos. And so we already have the highlights for chapter 9. After chapter 9, we're going to go to this fourth section of books, 17, 18, 19, 20. Those are chapters that lead us into cancer biology and are the center of our course. And so we had that clay model video, we took the quiz. We're not going to do the assignment, but the assignment was to physically work with this stuff. It's a physical thing. People are deciding about the 4-H STEM Ambassador Program, and if you have any questions about that, you can contact Sarah Sparks. Again, the link to the program and application are in our syllabus, which is on Brightspace, and Sarah's email is there too, so you can just click on them there. There's that training schedule that's part of their website. So we need to decide by Tuesday the 7th and participate in the training session if we're going to do 4-H STEM Ambassadors. And that's that. All right, so here are these 3 sections to chapter 1. When I see those sections, I say, oh, those first 2 sections, I would talk about those the same way. And it's different than our authors. And of course, I think it's a better thing. Fundamentally, when you wanted to talk about the origin and evolution of cells, I would say, the way we understand those is through model organisms like that. That's the best way we understand those, and then extend out into the broader diversity of life on Earth like that. But we just have a tremendous foothold on the explanation of evolution. The other wants to talk a little bit about the origin of life, and that's really murky, and it's kind of always going to be really murky. It's speculative about what happened, but there's a reason why the other wants to talk about it, and I'll get to that reason with it, approaching it a different way. And so, how would I approach this? I would say, well, let's define a gene, okay? Because that's what we're going to be talking about. When we want to talk about a model organism, I've said, we'd be talking about related genes. That's what we're talking about, It's not that the organism can grow in a lab and reproduces quickly. Those are secondary reasons. The fundamental reason is, they're really closely related. So those are model organisms. We can understand about ourselves or other organisms in studying a small number of model organisms and then reaching out into the diversity of life on Earth like that, okay? And then the next thing I'd say is, I would want to show students this. If you want to talk about evolution and you want to talk about a model organism, I would start out like this, and I would say, we talked about this a little bit in class, right at the end of class last time, and it's this thing where we take the DNA sequence information from one species and we search another species. And so That's called BLAST. The program is called BLAST, and so we just say that's the verb. We're going to blast, and it's like a word search, looking for the word, library in your term paper, and you just ask the, word to search for library and find it. And so that's what BLAST does. It's also like a Google search where you get hits, and both BLAST and Google rank them from highest to lowest. And so that's what's happened here. And what I've done is, well, we've been talking about myogenin. So I took the human myogenin amino acid sequence, the protein sequence, and I searched it against just the domestic dog genome. And that's Canis lupus, and actually the highest match it shows is from a dingo. And but it shows it can find a sequence very similar to human in dog. And we're not surprised by that, but what I wanted to emphasize in class is what we should be surprised at is this number, and it's the probability. It's the likelihood that the dog and human myogenin amino acid sequence would be that similar if it was just a chance event. If we were just picking amino acids randomly, what's the chance that the human and dog myogenin gene would look so similar? And this is a fundamentally, profoundly significant concept, and that is that number says, oh, it's not a 1 in a million, that'd be 10 to the minus 6th, or a 1 to a billion, that'd be 10 to the minus 9th, or a 1 to a trillion, that'd be 10 to the minus 12th. It's a 1 in 10 to the negative 115. So a 1 with 115 zeros. That's the chance. It just couldn't happen. It's never happened. Something that rare, I'm going to claim, has never happened anywhere in the universe, in the history of the universe. That's just unbelievably rare. We only have one explanation, and the explanation is that we'd say we think that they are, this just shows the amino acid similarity. They're very, very similar. And that's our interpretation, that they must have shared a common ancestor, the dog and human, that already had the myogenin gene, and it stayed really, really similar. That we're not looking at convergence, we're not looking at these becoming similar by chance, but that they becoming slightly different by chance over evolutionary time. And in this figure I made, I used a different gene. We could use it for any gene when we want to compare between species like that. And that ortholog is based both on that astonishing sequence similarity and then also studying it, right? We also need to study the myogenin in dog and humans and say, yeah, it's doing the same thing. It's a transcription factor that activates muscle genes during differentiation. So that combination makes this real strong claim. Lots of scientists are reluctant to use that because we'll never be able to prove this, because we'll never get that ancestral gene. It's just a long, long time ago. And we can find some old DNA like that from tens of thousands of years ago. That DNA is really damaged DNA, and it's just really possibly unlikely that we'll ever be able to get back to DNA from something that's that long ago. So instead of saying orthologs, a lot of scientists would like to say homolog, and a homolog means really similar, but it doesn't make as strong a claim as that. We're going to say ortholog a lot because the evidence is just astonishing, but just be aware that some scientists use that term, okay? So we see this astonishing sequence similarity. We see the protein myogenin doing the same thing in dogs and humans. So we say, okay, solid, that's an ortholog. Here's the next thing. Here we are blasting the human myogenin against the fruit fly genome. So that's Drosophila melanogaster, the fruit fly, and other model organisms. So model organism because we find astonishing similarity between myogenin in humans and fruit flies. And when studied, it does the same thing. It's a transcription factor that binds the enhancer of muscle genes and activates them during muscle differentiation. Fruit flies have muscles. They walk around, they fly around like that. And it appears that our last common ancestor, which in this case, between humans and fruit flies, was about 600 million years ago, last common ancestor had myogenin. It was like a little worm, wimpy little worm, that was crawling around with some wimpy little muscles like that. And that's been retained, okay? So that's that. And we can do that blast search, and we can find that, and we can do that for lots of different genes. not every gene, but lots and lots of different genes. And that's the fundamental basis for saying we're studying a model organism. It's because it's astonishingly closely related. That's still an astonishing number. What is the chance that those would be that similar? That's 2 times 10 to the minus 20th. 2 times, with 20 zeros, right? That's never happened, too, right? And so it's really easy to do that, right? Now let's look at this. I blasted the human myogenic gene against, does it show it on here? Saccharomyces cerevisiae, which is the yeast that causes bread to rise. And that's another really powerful model organism, because we have lots of orthologs with yeast. How about myogenin? No, yeast don't have muscles. Right? So we can find myogenin because it was evolved early in the evolution of animal muscles, and has been used over and over again. But yeast didn't have it yet, and so it didn't find any similarity. So we can switch to looking between a human protein and a yeast protein. We can switch to something we'd expect yeast to have. And something we'd expect yeast to have is similar regulation of the cell cycle, because the yeast is the same kind of cell, talked about in chapter 1. It's a eukaryote. And so we have a lot of things that are similar between us and yeast, but they're not at the tissue level, not at things like muscle and brain cells, but down at that single-celled level, we see that similarity. So we're going to talk about the cell cycle in chapter 18. So we're heading there pretty quickly. That's one of the 4 chapters in the 4th section. Chapter 17, 18, 19, and 20, I think, are the numbered. And so we're going to get to the cell cycle pretty soon, and we'll see a lot of similarity between that and yeast. And so I just picked one of those, and I picked, you don't need to know it, but it's a human cell cycle regulatory protein, CDC25, that we're going to learn about in chapter 18. And I said, let's blast that against Saccharomyces cerevisiae, the yeast that causes bread to rise. And we find this, right? 7 times 10 to the minus 28th similarity. There's a lot of differences. Look, only about 1/3 of the amino acids are identical. But it's based on these 2 things. One, that that proteins are long, hundreds of amino acids long, so we have like 100 comparisons that are being made, hundreds of comparisons being made, and then every comparison is this 1 in 20 chance. You are picking 1 out of 20 amino acids, you have a 1 in 20 chance of matching. So that the probability of that happening goes down very, very fast as you find more and more matches. So even though they are only 1/3 similar, they still show astonishing sequence similarity. And yeast becomes a model organism. The scientists that discovered this protein and others in yeast that cause bread to rise won the Nobel Prize in physiology or medicine for this reason, that human orthologs of these also occur and are essential in us in regulating the cell cycle. You can look for CDC25 in single-celled yeast and find it. What does that say? That also says you can look for it in any animal and find it. You can look for it in any plant and find it. However, if you look in bacteria, which are prokaryotes, and you blast a prokaryote with it, you can't find it. Because the bacteria are single-celled, but they don't divide in the same way that eukaryotes divide. And eukaryotes divide by mitosis, and so in chapter 18 we'll learn about the mitotic cell cycle and regulation of it. Lots and lots of orthologs in all plants, all animals, and in single-celled eukaryotes. Because single-celled eukaryotes evolved the capacity to regulate the cell cycle like that. Bacteria divide differently, they divide by fission. So if we blast CDC25 against an even more distantly related species, a bacteria, we won't find it. But if we go to some other features of life that we share with bacteria, we can find it. So in this last one, I said, let's take E. coli. That's a powerful model organism of bacteria that scientists study a lot in molecular biology and is naturally found in our digestive tract. If we blast a human DNA polymerase sequence against E. coli, we find a match. So this is, let's see, is it written out here? No, not fully. It's a DNA polymerase because every cell on our planet has DNA. And when it's time to replicate the DNA, it needs a DNA polymerase. And so we can find evolutionary relationships between us and something that's profoundly different than us, is the little bacteria in our digestive tract. And we can do that by searching for orthologs or homologs, if you want to be, more gentle about the claim, orthologs of genes that we would expect to have in common that need to code for common proteins like DNA polymerase. And there's that number, right? Still a similarity of about 38% or about a third of amino acids, and that gives us this astonishingly low probability. So that's the key thing when I want to talk about evolution, and then I want to talk about model organisms. I'm like, that's the thing I want to talk about. It's just astonishing. Those numbers are astonishing, and you can do that for thousands of genes like that. One way to think about it is this. One way to think about it is that life, like here's a human, and then they reproduce with sperm and eggs like that, and then here's a human. So this could be your grandparents, and this could be your parents, and this could be you, right? And that's the history of life on Earth. The history of life on Earth is there's one generation after another. And that's true for organisms like plants and animals. It's true for single-celled organisms like yeast and bacteria as well, right? It's nice to think about it for like a human just because it brings it home, right? So there's a history, right? This is a history. It just goes on and on, back in time, right? And we're talking about the last common ancestor, like your super great-great-grandparent that was shared between you and a dog, or you and a fruit fly, or you and yeast, or you and bacteria, like that. We just go back a generation. So we're looking at that timeline, and what we're used to thinking about is we're used to thinking about the organism on the timeline. And we talk about the evolution of hair, or the evolution of a wing, or a limb, like that, right? Or a brain, like that, right? And we're thinking about the organism. But there is this feature of life on Earth where we go from one generation to another through a kind of little bottleneck, where the most prominent feature of this path is, yes, the organism, but then in every generation, a curious little shift from organism to mostly DNA, right? What did we inherit from our parents? Not an arm. We inherited the DNA to code for an arm. And scientists can collect this information, and computers can happily store it and analyze it and find these comparisons that are just astonishing. And we can paint the story of life on Earth like we always have done, looking at the organism, or just repaint it and just say, we're not even thinking about the organism. We're not going to forget it, but we're just going to focus on the DNA sequence similarities and just go with huge amounts of data. What's the evidence for the history of life on Earth? Oh my gosh, right? It's just astounding amount of data. So I would really want to emphasize that. What are we talking about here? And this is something else that some of this stuff that I teach, I just am kind of, making it up. But this is a really good analogy. This is some sand. And the statement is evolution is both a process and a pattern. And so what we've just been talking about is the pattern. That evolution occurred and is occurring, and that's a process. And then it leaves a pattern. So here's the analogy. A snake crawling through the desert is a process, and its trail is a pattern. Okay? So that's, both of those things are true about a snake crawling, is that the crawling is a process and it produces a pattern. And that's what evolution is like. Evolution is a process and a pattern. And here's what we have to imagine, is just that the snake crawls through the desert at night. Evolution occurred over millions of years ago. We didn't see it. It was like nighttime. And so we didn't see the process. But let's say we walk through the desert in a day, and one of us says, hey, my grandma taught me that this is a pattern, that if we follow it in that direction, just based on the shape of the marks, that will come to an oasis. No, follow that direction, because that snake is heading toward an oasis, right? Like that, right? So that snake is going to go find water, and if we follow the pattern, we'll get to the oasis. And so this could be life-saving. And that's true of evolution too, that the evolutionary pattern has been followed with the understanding that it's a process that leads to something, and that process is the evolutionary history that leads us to the opportunity to, say, study model organisms because they are related like that, and make life-saving discoveries, right? Each of us have been, our lives have been saved many times by scientists studying the pattern of evolution like that. So I say the process of evolution is covered, right? We didn't see the process. We really are focused on research of the pattern of evolution. The fact that the hands are covering that means that the theory of evolution is always going to be a theory, and that's why. This occurs and occurring over millions of years. Humans will never ever directly observe the long-scale evolutionary changes that have to occur for macroevolution, for big changes to occur in evolution. So theory of evolution, it's always going to be a theory like that. It is always going to be a theory, and that's fine, right? I don't have a problem with that, right? The pattern is not a theory. The pattern is the opposite. The pattern is like, as obvious as the, skin on the back of your hand. The data in the pattern is just, just growing and abundant like that. And so that's part of evolution, and we talk about the pattern. Everything in biology is about the pattern. The DNA sequence is part of this pattern. The pattern is a product of a process, and the interpretation, seeing those similarities, is orthologs, that over evolutionary time, that's what's occurred, like that. There are lots of examples of using DNA to analyze evolutionary relatedness, like forensic testing in DNA, right? People get convicted and go to jail, prison, because their DNA matched a sample at the crime scene. Then paternity, we have no problem testing, who the father of a child is by just examining the DNA, and the evidence is overwhelming because of the pattern. The data in the pattern is so big like that. And this is what it looks like. Computers could just line up orthologs across many species, right? We looked at, humans, dogs, fruit flies, and yeast like that. Well, that list can just go on and on and on, And so if we just do one, we just look at one like myogenin, the evidence is astonishing. And then you say, you want to do that again? You want to do it on another gene? Because we can. And it's just the same story over and over again for all the genes like that. We see, so there aren't orthologs because cells don't have it. Yeast don't have muscles, so there aren't orthologs in myogenin in muscle. And all that pattern that is so much data, all points to the same thing, that there was this evolutionary process. I mentioned on the first day of our class, bio's like a three-legged stool, and the stool itself is the pattern of evolution. That's why I said the stool is evolution. Evolution is a process and a pattern, and everything in biology, every possible example, every possible observation, every possible piece of data is part of the pattern of what appears to be an evolutionary process over hundreds of millions of years. This is a pattern. Everything about this, not just the myogenin, but the whole story of the pattern of cell differentiation, of the steps in cell differentiation, is a pattern like that. Thus, model organisms are an example of a pattern. The fact that we can use them is part of this pattern story, and that's why we study them. They're cute and they grow fast and they don't eat much, right? Like that. But the key thing is the evolutionary relationship, like that. And you're asked about that on an exam, you gotta say that first. So we really are talking about the pattern when we're doing those DNA sequence similarities, but everything we're talking about is this pattern, and it's important to appreciate that. And then, just before we leave this process is not completely covered up. So just to go back to that, everybody understands microevolutionary processes. the development of dog breeds or modern plant species that we grow for agriculture, that involved selection through artificial selection and microevolutionary change. The COVID virus is evolving rapidly. Those variants are all the result of evolution through microevolutionary change. Everybody knows you have to take an antibiotic until they're all gone, or the bacteria might evolve in your body and kill you, right? So everybody understands, because you can directly prove microevolutionary processes. So in a sense, we can see the snake wiggling its tail. Definitely that's been happening, right? And what's still covered up is just the real macroevolutionary changes, the really big changes that take hundreds of million years to occur. Again, that's always going to be covered up. But we also have that partly uncovered, because this is where the fossil record fits in, right? Is that we have direct evidence of what kind of organisms lived back then, and the fossil record is improving every day. It's a miracle to imagine. What do you think the chances are that you are going to get fossilized, right? That somehow, like a bus is going to run you over and they pave the road before they notice your body, and so you get encased in tar and your body gets preserved for hundreds of millions of years. You get turned into a rock like this little guy did, right? Yeah. So it's a miracle that there are such things as fossils, but the fossils fit in, and they further uncover this. It's still covered like that, but we see, right, we see some of the evidence of what's happened in the past by direct history of organisms like that, or this guy. And the fossil record keeps getting better and better. This is a fossil of a mammal that was a four-legged land-based mammal that was starting to spend more time in the water and on the evolutionary pathway to marine mammals, whales and dolphins. And the fossil record for that has got astonishingly good, right? There used to be gaps and best guesses, but just more and more fossils get found again, not that frequently, because they're just so crazy to imagine that any exist at all. But we have the fossil record, right? And so I would want to emphasize that about this, is that, we talk about a process and a pattern, and the pattern is all we're going to talk about. That's that DNA sequence analysis, but it's everything else too. It's just that DNA sequence analysis stands out because of the numbers, because of the data. The way the data was organized and how a computer can sift through it just makes that a huge amount of pattern evidence, okay? So we get something like this shown in the book, and it's just showing, hey, here's the evolution of life on Earth, and the evidence for that is great. And it used to be like, well, these animals all look like they have hair, so we're going to put them in the same category, right? And so we were focusing on the organism part of it. But then there's this step through the history of life on Earth when the story was mostly DNA. In every generation, it goes from, it's all about the organism to, it's kind of almost all about the DNA, like that, okay? All right. And so just a little bit more on what the book specifically said. The book talks about, first cells and stuff like that. And these are really good things, and you're required to read them, but, not something that I'm like, I really want to talk a lot more about, origins. We can answer questions about it in class or after class. I just want to show you this. I've mentioned before that Intro Bio, as taught anywhere, is really this course, and it's just evidence of just how dramatically these topics have, taken over biology. Okay, so I just want to show you this. It's not the 7 themes for our course. It's the 7 themes for this course I teach in Intro Bio. I say all living things are composed of cells. All cells come from pre-existing cells. That's called the cell theory. That emerged in the 1850s. Part of the cell theories, naturalists thought that maybe something like spontaneous generation could occur, and you just get life from nothing. Okay, so our book is introducing this. It's cell biology for a reason. And then all cells exist in a water environment, and all cells have really similar chemistry. They're surrounded by a membrane, a cell membrane that's a phospholipid bilayer. Every cell on our planet. Bacteria, right, our most distant relatives, have a really similar cell biology characteristic. And then really similar chemistries. Every cell on our planet needs to make its own proteins. and those will be species-specific proteins: dog proteins, human proteins, fruit fly proteins, yeast proteins. DNA is the primary heritable molecule, and we know that there are coding genes and non-coding genes, but the big job is to code, that have the storage information to be output and code for all the different human proteins and all the different dog proteins like that. And then the final concept is this concept of gene regulation totoponency. And you can see we're already emphasizing that in this course. And so our course is really similarly structured to that. And you don't need to memorize these. I'm showing them because we're talking about, the structure of our course. Yep. So then I just want to talk a little bit about model organisms like that. So here's some classic model organisms. We have mice like that, laboratory mice, and fruit flies. And here's one that's a little worm. It's called a nematode. And we probably won't use examples of nematode very often, but they could pop up and just know that it's a simple invertebrate like that. Then here's Saccharomyces cerevisiae. Again, it's the yeast that causes bread to rise. And there are lots of different species of yeast, but that's the one that's really the model organism of yeast, which is a single eukaryote. Here's an interesting one that's a very tiny weed called Arabidopsis thaliana. Arabidopsis is what people just call it, and it's like the fruit fly of flowering plants. And then I mentioned E. coli. So here's E. coli growing like that. It's a bacteria found in our digestive tube, and scientists study it. And then here's an interesting variation of that. This is where they spread the bacteria out on a plate. Here's where they just took the bacteria and they swirled it in with some melted agar and poured that on a plate. And so this is called a streak plate, where you streak it out. And then what grows here is referred to as a lawn, because it's just billions and billions of bacteria just smoothly spread out across the plate. And what you can see here are some holes, little holes like that, where there aren't any bacteria. So we have a lawn, and then cells missing in these little things. And the little things are called plaques. You can see more here and more here. Almost all of the cells are dead because of these plaques. The plaques are where the bacteria are missing, and the plaques are there because of a virus. And so that's another model organism. Viruses aren't alive, but they're an organism, and we'll learn some about viruses, because scientists started molecular biology down at this very minimal level to try to understand the organization of virus, and they especially tried to understand organization of bacterial viruses because they're such simple organisms. Both the bacteria and the viruses are simple. And so just some pictures of some of these that we'll talk about and learn about. Here's a little picture of a nematode. It's a video, and I guess I just didn't have the video running. And fruit fly like that. And then another one that was not pictured on my first is a vertebrate called a zebrafish. And the zebrafish, this is what's called a transgenic organism. Transgenic means genetically modified. We took a DNA and put it into the organism and into its DNA, into its chromosomes, and the gene we put in is called a transgene. If you buy food at the store, it's referred to as GMO. That's the same as transgene, genetically modified organism. Here's a little transgenic zebrafish, and it's glowing because a gene from jellyfish was put into it, a transgene, and that when expressed as a protein, it glows green. So the DNA doesn't glow and the RNA doesn't glow, but the protein product glows. So anytime and anywhere that gene is expressed, it will glow. And what they have here is a transgenic zebrafish that has the GFP transcription unit, right? The business part of the jellyfish gene that can be expressed. And what they did is, in a test tube, they linked it to the regulatory region. And in this case, it would be the regulatory region from a zebrafish. And it would be a gene that's normally expressed in the nervous system, in neuron cells that are part of the nervous system. So this is just a regulatory region from the gene, from a zebrafish gene expressed in the nervous system. They attach that to GFP. They put the transgene back into the organism, and we have a strain, a variety, a breed, you could say, of zebrafish that has the nervous system going green, like that. That same strategy is done in other organisms, and one thing that's really powerful is that in mice, now scientists are studying organs. So this is the development of the kidney. And we have model organisms in cell microbiology, and now with these powerful tools, we're starting to have model organs. So there's some organs that are being studied that are model organisms. The kidney is one, because people are desperate to have new kidneys, because kidneys don't heal. There is no such thing as a stem cell to help a kidney heal. And when folks get kidney disease, it just means their kidneys have been damaged and they're not working, and they have to go on dialysis to get help like that and wait for a transplant. What about learning about the kidney and growing a kidney, right, in a dish? And these are would be called model organisms. Then another one is that cells can be growing outside the organism. And so here's a flask, and it has nutrients, and then it has millions and millions of cells. They're just dispersed cells. They could be floating around, or they can be stuck just on the base of the plastic in the dish, and they're growing. And they could be human cell lines, or they could be blue whale cell lines, and that provides another powerful way of working with cells like human cells without hurting a person, or blue whale cells without catching a blue whale, like that. And that's another powerful tool in molecular biology. And sometimes people say that this is being done in vitro, and that means outside of the organism. And in the organism is called in vivo. And so molecular biologists, cell biologists study a lot of cell lines because they want to study cells and molecules, and they can do that under very controlled conditions. This just fits in with really that earlier slide of looking at different tissues and considering those to be model organisms, like that. Okay, all of that is rooted in this concept of orthologs. And again, if you're reluctant to call them orthologs, homologs, but the evidence is just astonishing for this interpretation and this interpretation of the pattern of what we see, the evidence, what we see. Okay, and so I just made this in this first chapter, really talking about cell biology, and we're talking about, yes, viruses, which aren't cells and aren't organisms, but then all these, we got prokaryotic cells, and then we got single-celled eukaryotes, we got multicellular eukaryotic cells. So we're learning and relearning some of our stuff from intro biology, just about cells and the cell theory, like that, okay? So another thing is this idea of viruses, and we'll talk about some about viruses in our course because they're used as a model organism. They're also studied because of what they do. And the most common type of virus that we're going to talk about is our retroviruses, and we'll explain how they fit into our course. And here's just an example of the name of a particular retrovirus, and you don't need to know that name, but we'll talk about it. And then there's some other viruses that we'll talk about in our chapters on cancer, because, for example, these DNA viruses are found in certain cancers and are associated with helping to cause the cancer, like that. And so we'll learn some about viruses. This is a virus. This is the virus that caused the plaques on that dish of E. coli bacteria. And so this was the first model organism in molecular biology back in the 1940s and 50s. Scientists were really picking on this, and it literally physically looks like this under an electron microscope and based on evidence of the structural proteins. And viruses that infect bacteria are called bacteriophage. Yep, okay. And so then that's me wanting to talk about those kind of things. I want to talk some about the book, but I really would want to emphasize something different and parallel, and that happens some in our course. Okay, so then this third section, notice it says cell biology. So remember, our course is really two courses in one, cell biology and molecular biology. And after we read chapter 1, that is going to be chapter 4. And chapter 4 is called the fundamentals of molecular biology. And so, in a sense, we can think of this section 3 as introducing some features of cell biology, and then chapter 4 introducing some features of molecular biology, right? And it's good to look at these, right? Because I'll look at these and say, okay, well, you know, I didn't talk about super resolution microscopy, but there it is. It's considered to be an important thing, right? And there are these highlights like this. And remember, we have 2-point questions. So, you know, well, what is super resolution microscopy? You know, what is it used for? And so, you know, 2 points to explain that, right? And look at that, compare electron and light microscopy, right? 2 points to do that, right? And so there's stuff for you to get out of this chapter that this is guiding you to. Then look at the section headings and the bold terms and the figures to identify the highlights from this chapter. I'm not going to, this section, I'm not going to talk about it very much, but in cell biology, we're looking at cells. And so we're looking at cell concepts and topics that you would not know if you just looked down at a molecular level, like the cell membrane and understanding the function of the cell membrane, like that. We want to make sure we understand, you know, prokaryotic cells represent bacteria and you know, a few other cell types, and then eukaryotic cells are the ones that have lots of organelles and are the ones that make up plants and animals, but also quite a few single-celled eukaryotes like yeast. Okay, here's just a little analogy, a scale analogy that I like to talk about, that we'll talk about some, and it's like this. It says, here is an axis in the metric system for the size of cells, and we're talking about cell biology, and then down to the size of molecules. And so that's the range we're going to be in our course, like that, ranging from about 100 microns down to 1/10 of a nanometer. And I say, just imagine that one cell was the size of this region of Boston. And I know, I didn't grow up around here, but just a big city with suburbs out like that. And if that's about 30 miles across, then we could put this scale over here. And on this scale, if this was just a typical cell of yours, you could see that something like mitochondria is a mile across. Something like mitochondria is like a small suburb, right, like that. So pretty big. Then drop down to here. In this scale, physical scale like that, something like a water molecule would be the size of a basketball, like that. And then bigger molecules getting up to bigger sizes. A protein would be the size of like a truck, a big truck, and a range of sizes depending on the protein. And then we get up to viruses, and ribosomes are bigger. A ribosome is like school, right? Like a high school. And viruses get to be like the size of a block, like that, right? So it's really good at, you can maybe, save a screenshot of this as a reference when we talk about scale. Because these are tiny sizes and it's hard to keep track of the size, like that. Over here, I'm just showing that if you pinch your fingers together like that and hold them as steady as you can, that's about a millimeter, that gap, and you could fit about 20 cells. So a cell is too small to see. A typical human cell is too small to see, but not by a lot. And if we could see it, we'd see something like Boston with a lot of activity and activity on these kind of size scales. And that's for sure, right? The small stuff we're talking about is physical and real. And in this chapter, I'm not going to take time to talk about some topics because the book does a good job just explaining them like that. And so if you're confused about some explanations like numerical aperture, um, right, a question on the exam could be what role does numerical aperture play in a light microscope? Right? Like that, and try to get from the book two things that are significant about numerical aperture. Then here's another thing, just another approach in cell biology, right? Oh, I should say, cell biologists love to look at microscopes and light microscopes because cells are small, but not really small. So, light microscopy is a really powerful tool. And our book talks about a variety of different types of light microscopy like that, okay? Then here's another powerful tool, and that's just to take a sample and put it in a tube and then spin it in a centrifuge. And we can start separating cells from other components, and then we can tear the cells apart and separate them more. Let me just give you an example. I don't know if I have a picture of this here, but I'll just show you an example. So here's an example of some cell biology type of method, and it's one that I don't think our book gets into. But there's a glass, it looks like an old-time piece of lab equipment. It looks like that. It's a test tube that's got a wide part here. And there's a plunger that fits in the test tube. And so it goes down in here, and what you can do is you can put liquid in here. It bulges out here just so the liquid doesn't splash out, because what you're going to do is you're just going to move that plunger up and down in this tube. And this thing is called a Downs homogenizer. And there's a gap between this glass ball. All this is made of glass. Like this glass ball fits in here pretty tightly, but there's a gap. There's a gap between that glass ball and the wall of the tube. And that gap is very specific, and the gap is less than the diameter of a cell. But it's greater, for example, than the diameter of the nucleus. So we can do something called a nuclear extract. We can isolate nuclei from a cell by putting the cells in liquid and then using a Downs homogenator to homogenize them, and it will just physically tear apart the cell, but not the nucleus. And after we have done that Downs homogenizing, we can put the cells in a test tube and we can centrifuge them, and the nuclei will be in the pellet in the bottom because they're intact and dense and heavy, and they will pellet. The centrifuge will spin, increase the force of gravity, essentially, and the heavier molecules, heavier materials will be pulled down. And the rest of the stuff, like bits of the cell membrane and the ribosomes and so forth, will be up here in solution. And so the term for that classic centrifugation would be that we have a pellet of nuclei, and then this is called, the liquid is called the supernatant. The supernatant is garbage. We want to save the nuclei and throw away this supernatant. And that's a good example of the use of centrifugation, is to isolate nuclei and study nuclei, because the genes are in there and the transcription factors are in there and RNA polymerase is in there. So we'll talk about making a nuclear extract pretty often in our course. Awesome. Okay? And then, so just an example of centrifugation is isolating nuclei. And our book talks about it. You can isolate other, you could isolate the ribosomes or the mitochondria and study them, right? And so that's a part of cell biology, right? Is look at all that stuff in the cell. Can we separate them and purify them and study them? And then there are other techniques that are described, and we can talk about in class if you have questions about them. Yep. All right. So now I'm going to move on to chapter 4. And again, I talked about chapter 1 quite differently, and I think that's great. Bring questions to class that you have to clarify how the book talks about chapter 1, because we want to learn both those. The same is sort of true here for fundamentals of molecular biology, and we're not going to talk about everything, but you're required to read the chapter and look for stuff that's important. the most important stuff. And that's what you want to learn from this chapter, and bring questions in about stuff that you're confused about, okay? And so, see, fundamentals of molecular biology, well, we're really rolling on this, right? So messenger RNA and genetic code and so forth like that. Something that I do want to talk about is this retroviruses, because that's going to be a central part of our focus on molecular biology. And then we're going to go to chapter 9, and that's going to be about transcription and gene expression, so, you know, the DNA models. Then we're going to go to chapter 17, and that's going to be some cancer biology. And these retroviruses, a certain set of retroviruses, really help scientists get going on the study of cancer and using molecular biology to power the discoveries like that. So this is an important chapter like that, and we're skipping chapters 2 and 3, which are about chemistry, biochemistry, and metabolism, because you've learned some of that, and we want to emphasize some other stuff in this course like that. Okay, so molecular biology, and one of the things is traditionally molecular biology is the study of the biochemical study of DNA as a physical molecule, and then the primary heritable molecule. I say primary heritable molecule because in between organisms, right, from your parents to you, DNA was the main story, but there was a sperm and egg. There were cells also, right? The cell theory says that we have cells from, we come from cells. We come from cells, but in those cells, in the sperm and egg, the big story, primary story is that the DNA holds a lot of information. So here's what is, it used to be told humorously, like when molecular biologists first start to say, oh, this is a central dogma, they meant it as a joke. And I don't remember why it's a joke, but it's stated this way. And we're already focusing on that, right? The central dogma of molecular biology is about gene expression and like the expression of myogenin in muscle differentiation you're already learning about, okay? Like that, okay? So we've already learned a lot about this, and again, this is fundamental stuff in molecular biology. It's also stories from chapter 9 that we're heading toward, okay? So here's what I want to add to that, start adding to that, and that is that we've already looked with regard to gene expression and talked about transcription factors being the dynamic, pivotal story in a lot of gene regulation. And not all of it, and we'll learn parts of it, but if you said, I want to introduce molecular biology, the fundamentals of molecular biology, and what you'd say in about the third sentence is, oh, molecular biologists are in love with transcription factors and how they're regulated to regulate gene expression. It's also nice to pick on transcription factors because they're all proteins, always, they're proteins. And we learn a lot about protein regulation just by picking on transcription factors and learning different stories about them, okay? So here's this FOXO, right? And this slide I didn't show, but this is when there is a growth factor and this cell is responding, and it's responding to blocking FOXO, keeping it out of the nucleus. If the growth factor is not present, then FOXO does not get phosphorylated, does not get sequestered by 14-3-3 in a cytoplasm. FOXO goes into the nucleus, it binds to enhancer, and it activates about 200 different genes, the expression of about 200 different genes. And its function is to slow aging. Fascinating, right? Like that. For us, it's just look at that molecular switch. So we have this thing, a big picture feature of our course is to learn about transcription regulation and focus on transcription factors, which are proteins that bind to enhancers, and we're going to have a list. So we have a list of MyoD, and it just gets made. We can add myogenin to that, because that's another transcription factor we learned about. Then FOXO. We just touched that a little bit. We're going to say, oh, look, we learned from FOXO two things. That it's going to be excluded from the nucleus, and it's going to be phosphorylated, okay? So that's that, okay? The next thing is just to say about this that we just haven't emphasized is I'm going to redraw this and zoom out just a little bit, okay? And so this is like what we're showing here is a cell, and this is what we would call a target cell, and it's a target of this molecule, and in the example figure, it's a growth factor, And so that's a molecule. It could be a growth factor, and in general, we could call this a signaling molecule or a signaling factor. And there are lots of different categories, and in different areas of biology, a different feature is emphasized, and sometimes there's some different terminology. So like in neurobiology, when cells communicate with each other, they communicate it with a neurotransmitter. So all neurotransmitters fit into this general concept of the signaling molecule. Or in immunology, our immune system cells get activated at the site of infection by releasing molecules that are signaling molecules called cytokines or chemokines. Like interleukin is a famous example of that. And then all hormones fit into this. All hormones go between cells and bring a signal, a message. And hormones are also synonymously referred to as endocrine factors. And the big field is endocrinology, the study of hormones, is all about the study of these signaling molecules. So we have signaling molecules, and they're binding to a target cell. And there must be a cell that made those signaling molecules, whatever they are, and that's called a source cell. We're really interested in this target cell. And to respond, the target cell is going to have a receptor. And really typically, the signaling molecule doesn't come into the cell. It stays outside of the cell, and somehow it's binding to this receptor, activates the receptor, which are always proteins, and then the receptor communicates the presence into the cell through a path, a biochemical path called signal transduction that we'll learn about in chapter 17. And then there's going to be this target cell is going to make a cell response. So that's what's shown. In the case of FOXO, it could speed up or slow down its rate of aging, like that. Okay? And so this is really common. Chapter 17 tells the story of this, and we're heading toward this. And just to add this, when Riley comes in, he's going to talk about a cell response that is our immune system cell. So this is going to be an immune system cell for fighting infections. In this case, it's going to be for fighting and killing cancer cells. So it's an immune cell. It's going to make a cell response to get activated to fight this cancer cell. And it's going to do that by having, in Riley's case, the receptor being something called a CAR receptor. So that's Riley's story right there. He's going to come in and he's going to talk to us about how scientists make a special modified transgene called a CAR to put into our immune system cells so that they'll be better able to respond and fight cancer. And so this drawing is as fundamental to our course as the clay model videos. This drawing of a signaling molecule binding to a receptor and a signal transduction pathway leading to a cell response. Next thing is, in this cell response, there's lots of different stuff can happen, okay? And they can fall into two categories. And these terms are uncommon, but it's a very good explanation. So there could be, in the cell response, there can be genomic responses. And genomic responses are in the nucleus, and it's changing gene expression. So the cellular response can include changing gene expression, right? And the other is called the non-genomic response. And that's where the cell's responding, but it's just not about gene expression. So it's responding in other ways, changing shape, migrating, and so forth like that. Lots of different types of non-genomic responses. But just understand that. And we're going to talk a lot about genomic responses because the way the cell makes genomic responses is that this signaling molecule through the receptor and a signal transduction pathway is going to be affecting the specific transcription factor. And it's going to be switching the transcription factor on or off. So remember, we like transcription factors. They're always proteins. There's a couple thousand different ones in humans. We know some already, MyoD, Myogen, and FOXO. And we're interested in how they get switched on and off. And they get switched on and off a lot of different ways, but one big category is that these signaling molecules can cause genomic responses that lead to changes in gene expression by regulating a transcription factor. That's what's happening in this figure. Is this growth factor is affecting transcription. This growth factor is affecting a genomic response. So on this figure here, woo! This is, some of these switches are going to be ultimately due to signaling molecules. Okay, and that's a lot of chapter 17. Okay, and we're going to introduce that in fundamentals of molecular biology, and you'll see why. We're going to introduce that and have a sort of a favorite example of that. Okay, so let's see if I happen to have my slides lined up, and that's where we're at. Time to talk about that example. Yeah. And the example is going to be another transcription factor we're going to learn about called ELK. And ELK is going to be activated by a kinase that phosphorylates it. So here's ELK protein in humans. That's a funny name, I know. The ELK protein is not phosphorylated. And the ELK protein is a transcription factor. And it's inactive. And then a kinase, and in this case, this kinase is, so it's an enzyme that's going to add a phosphate in a chemical reaction. So it's going to add a phosphate to ELK, and ELK is said to be phosphorylated. And the kinase enzyme is specific to add it to ELK and regulate it, and that kinase is called ERK. Okay, all right, and we have to get ERK activated, and there's going to be a growth factor that activates ERK as a result of signal transduction pathway, and when ELK gets phosphorylated, it's active. And, like in our clay model videos, the ELK binds to an enhancer and activates target genes. And this is a growth factor, and it's stimulating growth because the target genes include a gene for cyclin, which in chapter 18 we'll learn is important in the cell cycle, regulating the cell cycle. So our example is going to be a good one to take us as simply as we can through the fundamentals of molecular biology with an enthusiasm of gene expression, that DNA makes RNA, makes protein, central dogma. Learning about that by plugging it into this specific example that is a kind of a minimal example to see the role in chapter 17 about receptors and signal transduction and cell responses, and then chapter 18, simulation of the growth of the cell and the cell cycle. So that cell cycle is one potential response that cells can do in response to a signaling molecule, and that happens in response to a growth factor. Okay, so that's what we're going to be looking at, and our book talks about that example too. And so here it is. So again, talking about really chapter 4 and the fundamentals of molecular biology by putting them into a context, and the context we're heading toward is chapter 17, and it is a pathway. And so here's this pathway, and it's going to show growth factor and receptor, and then this signaling pathway where ERK gets activated, and then ERK phosphorylates ELK, and ELK activates gene expression of genes like cyclins, so the cell will go through the cell cycle. Okay? And it's phosphorylated ELK that is active and binds and activates the cell. Okay? So here's our example, and we're doing great. And here's our specific example. So on your skin, when you get cut under the scab, the skin is healing, and that means the cells are growing and dividing, so they're going through the cell cycle, chapter 18, and they're doing that because under the scab there's a growth factor that's stimulating this. So this is the cell membrane of skin cells, epidermal cells, under a scab, and what's happening. So all this is happening under that scab. And the case of the growth factor, it's called PDGF. And that's a good name because the name is platelet-derived growth factor. Okay, like that. And platelet-derived growth factor, the platelets at the site of a wound, platelets release clotting factor, and that clots the blood, makes the scab. They also release PDGF. The PDGF doesn't go into skin cells, it binds at the cell membrane to a protein called the PDGF receptor and activates the receptor on the skin. And then this signal transduction pathway takes place, and the skin cells are stimulated to grow and heal. So this is kind of the most direct example we have, and it's a good example because it happens under every scale. Okay? Okay, and so here's what we're going to learn in just a snapshot, and that is this PDGF receptor is going to be in chapter 17 an example of a receptor called a receptor tyrosine kinase. And when it gets activated, it adds phosphates to itself. And we'll learn about that. And they think there are 58 receptor tyrosine kinases in humans. One of them is the PDGF receptor. Okay? We're going to learn about them because quite a few of the receptor tyrosine kinases are growth factors. And when we're talking about cancer, we would want to talk about cells that could, or genes that could get mutated and cause cells to grow out of control. And receptor tyrosine kinases can. There can be mutations that cause these to be activated even in the absence of growth factor, and that causes them to grow uncontrollably. A gene that can get mutated and cause cancer is called an oncogene. So some of these receptors are oncogenes. They regulate normal growth, but when mutated can cause uncontrolled growth. Okay? All right, so now then we're just going to circle the names of the genes, that the proteins that we're going to know. So we're going to know PDGF, that's a protein, PDGF, platelet-derived growth factor, that's what it does. Then PDGF receptor. Then the PDGF receptor is going to ultimately, it's going to activate this RAS. And RAS is called a G protein. And our author studied RAS when he was your age. And RAS is also a very famous oncogene. RAS is the most common mutation found in human cancers. Human cancers really typically have multiple mutations. Multiple problems have happened by accident to cause the cancer to be really growing out of control. And the most common one that's found is about a third of human cancers have a mutation in RAS. There's a reason for that too, and I'll just describe this now. RAS is called a G protein because RAS protein is always bound by either GDP, and it's inactive, or when the PDGF receptor or another type of receptor activates it, the GDP is let go and the RAS is bound to GTP. And then the RAS stays active, so now it's active with GTP, stays active until it randomly hydrolyzes, it randomly breaks GTP down to GDP. So the RAS binds the GTP. It's also an enzyme. It has a catalytic domain that's going to degrade that and turn itself off. That is kind of an Achilles heel because, you know, when you have a mutation, the most likely thing is to inactivate the protein somehow, right? But in this case, what we do is we inactivate RAS's ability to turn itself off. So RAS is kind of a vulnerable protein to get a mutation because you get a mutation that inactivates the enzyme and the RAS just stays on. It just keeps going. It's on too much, too long, and it keeps stimulating this pathway even if there isn't a growth factor, like that. So we're going to learn about RAS because it's famous and there's a thread of RAS going through a huge amount of modern molecular biology. So I'll just erase this and then keep going on this. So we got PDGF, and again, we're going to know that well. PDGF receptor. RAS gets activated, so RAS. And then we just quickly say the next ones. We just go RAF, MEC, ERK, and then ELK. And each of these is a kinase. Each of these is a kinase that activates the downstream protein by phosphorylating. So RAF is a kinase that activates MEC and by phosphorylating it. MEC is a kinase that activates ERK by phosphorylating. ERK is a protein that activates ELK by phosphorylating it. Why so many steps? And that's because the signal is undergoing amplification. We just have a few molecules of PDGF bound on the skin cells, and they amplify the signal inside the cell. So signal transduction is partly about amplification. And then the other thing that's about signal transduction is it's about what you could call checks and balances, right? So we don't want to make a mistake and have this cell start to grow unless it's a real appropriate signal. So there's lots of checks and balances, and more generally that's called signal integration. Sometimes there is more than one regulatory factor, and these pathways integrate, or you could say there is some crosstalk, that checks and balances or crosstalk. And so we just say that quickly: RAS, MEC, ERK, ELK, like that, those steps. And ultimately we get to ELK. And there's ELK, and ELK is a transcription factor. When phosphorylated, it binds this and goes on like that, okay? So that's our pathway and our cell response, and it's a genomic response, but also leads to the cellular response of going through the cell cycle. Okay, great. I think we got to stop here, and we'll pick this up again next time, and we'll talk about fundamentals of molecular biology to talk about, especially we'll focus on that RAS oncogene, right? Gene that can cause cancer, contribute to cancer when mutated. All right, great. So again, take notes, write down questions, bring questions to class, start reading chapter 4 this weekend like that, and next week we'll say now read chapter 9. So, we have to roll forward. Remember, I have chapter 1 and 4 on closed reserve in the library through our Brightspace. So if you don't have your book yet, you can read them, you can keep up. But then chapter 9 will be our next chapter, and I'm not going to be putting that on closed reserve. There's a limit to copyright infringement. So take notes. You do not have to send me pics of this today because we all missed class today. All right? Great. And we'll see Riley on